Hello students, welcome to Solve Our Classes. This is Dr. Sarkar over here, and today in this lecture we are going to discuss the calculation of pI of amino acids. pI of amino acids, also known as isoelectric point, is calculated using the Henderson's equation, which is obviously pH equal to pK plus log concentration of the conjugate base divided by the concentration of the acid. Out here we have been given the amino acid glycine, which is having no extra other charge, okay, or one can say extra carboxylic acid group or extra NH3 plus group. So for this the calculation is very simple, in which we will say pi is equal to pKa1 plus pKa2 divided by 2, which means it is 2.35 plus 9.78 divided by 2, and the value will come to be 6.07. Now, the pH at which a molecule carries no net charge is called its isoelectric point. Isoelectric point can also be calculated for charged amino acids such as aspartic and glutamic acids, in which the important dissociation constants are pKa1 and pKa2, okay, and we will not use the pKa3 over here in case of acids. The example given over here is of that of glutamic acid. So for the calculation of pi for glutamic acid, it will be simply pKa1 added to pKa2 divided by 2, and the value will come out to be 6.17 by 2. That would mean 3.09. That will be the pi. Now for other amino acids such as arginine, histidine or lysine, here we have been given the example of a lysine amino acid, in which the pKs that we'll be considering will be that of pKa3 and pKa2 for the calculation of the pi value of lysine. Okay, so out here what we will do, pi will be equal to pK2 pK3 divided by 2, and that will result to give us 19.60 divided by 2, and the pi will be 9.80. So the take home message is, whenever you have an amino acid having two common functionalities, as in the case of arginine, we can lysine, we can see it is NH3 plus and NH3 plus. CO minus is the uncommon group. So for calculation of pi, we will take the pi values of pKa3 and pKa2. Similarly, if you are given aspartic acid or glutamic acid, as in the case of glutamic acid, you can see the common group is the COO minus group. So we will be taking the pKa values of these two groups, okay, to calculate the pi for these kind of acids. That is the thing that one has to remember. If you are given non-charged amino acids such as glycine, phenylalanine or alanine, in those cases take pKa2 and pK1 of the NH3 plus groups and CO minus groups to get the pi values. Thanks for listening. Keep visiting our classes for other tricks to know biochemistry. Thanks for listening. Have a great day.